Hi everyone, how are you doing? I'm hoping you're able to see my screen. This is what you're going to be receiving on your Schoology homepage for Living Environment. I'd like you to, when you're listening to the PowerPoint, to go ahead and label in your notebook number 1, number 2, number 3, and so on. So all of those pictures will be discussing different types of lab equipment, and I'd like you to go ahead and identify what each number is. There's two ways you can do it. One, you can go ahead and add a text box and write it in on the actual sheet itself, or you can number 1 through 24 in your science notebook and go ahead and identify just by writing the name down in your science notebook. Now you'll be receiving, when I see you, either your cohort 1 or cohort 2, you'll be receiving this actual printout of this worksheet, since not everyone's going to have laptops to work from yet. So either way, 1 through 24, identify and label in your science notebook, and I'll call on each one of you to at least contribute to 2 objects, and maybe even to go ahead and describe a little bit based on the PowerPoint. Or you can go ahead and print out this worksheet on your own and have this labeled. So this is going to count as your classwork. You will not need to submit this on Schoology, but you will need to bring this with you on class, your flipped classroom. Okay, so let's go over lab equipment. So some of you might be familiar with some lab equipment, some of you not, but we'll all hopefully after this PowerPoint be on the same page. The first thing we have is a test tube. I'll also have these in class, so when I ask you, I'll hold the actual item itself, and I'll ask you what each item is, and then even if you can go ahead and describe the purpose behind having that item. So the test tube itself is going to be holding liquids. It could hold solids, but it's hard to get the solid out. So usually liquids or semi-liquids, but never used for measuring. And the reason being is that if you look over here, there's no lines, there's no little increments, and we'll see that when we look at graduated cylinders, there will be lines on those. However, sometimes in science labs, there are actual lines on there. But again, that's just to show you that you've done your measurement correctly using a graduated cylinder. But never, 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 never measure using a beaker or, in this case, a test tube. It's not accurate, just for holding liquids. Okay, this is what we really use in biology, which is a hot plate. And we will be using it when we're designing the labs. A lot of times hot plates will actually have a beaker on top. And let's see if I can go ahead and draw that. So a lot of times when we heat liquids, right, or test tubes, we have our beaker. And I wasn't sure if I could change the color. Let's go ahead and do that. We'll go ahead and put our test tube in here. So a lot of times people will say, well, how do we heat the liquid? They want to just put the test tube on there. Obviously, that's not going to work. So you're going to put a beaker, and you're going to fill it with water. Oops, I'm trying to get the color here. Here we go. So you're going to go ahead and you're going to add the water to a beaker. And then you're going to go ahead and put your test tube there. I didn't draw it very well, but the test tube is going to be pointing away from you. So then that way, when anything comes out, or if vapors do come out, they're not going to come towards your eyes. Also, if you're standing right here, right, you should be wearing your little goggles here. And that's a beautiful drawing. Right? So you're going to be wearing your goggles, and the test tube will be pointing away from you. Okay, really, really important safety tip. Okay. So then we also have a dissecting tray. We're not going to be dissecting in biology class. They've gotten away from a lot of the anatomy pieces where you have to go ahead and dissect, since a lot of things are virtual now. But we will be looking at some specimens in a dissecting pan, and there is like a tar or like a heavy wax. And again, I'll bring these in class. You can actually, see them visually in class, and they're going to be for pinning down specimens so that it's easier to do dissections in. Forceps sometimes are called tweezers accidentally. There's different types of forceps. These forceps are more often used in biology because they're folded like this, where you can go ahead and you can pull off tissue. But there's different types, ones with different shapes towards the end. so that you can go ahead and do different things with those forceps. But pretty much for lifting, moving, removing tissue, and they're really an extension of your fingers. So that's how they're used. So you don't use your fingers when you're touching specimens. You always use forceps. We won't be using these in science class in biology, but in chemistry you often do. And this is so that when liquids do or if fall on you, you do not stain your clothes, ruin your clothes. In biology, we have a lot of stains and dyes, especially when we're using microscopes. So this prevents any accidents where your clothes might be ruined. Of course, it's also there for protection if you're dealing with acids and bases, which is quite common in biology. But we won't be dealing with lab coats because we won't be dealing with any type of acids or bases that will be too harmful. Obviously, again, your hands, you need to wash them, but they will not be burning through your clothes. These are a little hard to see on the picture, but again, I'll be bringing them to class, and they're really, really small. These are cover slips, and cover slips are really important when you're viewing specimens underneath a microscope because it allows the specimen to lay flat against the slide. This is really important because when we're using microscopes, and you'll be using them in class, you'll be seeing that it's really hard to see a specimen that's too thick. It has to be thin. The layer has to be see-through, transparent. And the cover slip allows that adhesion to the slide to make it as small as possible or thin as possible, almost like squishing meat in a sandwich. That's what the cover slip does. It's like the bread on top where it squishes it down. These are dissecting pins. You will not be using those. And if you do see them, they'll be laid out on a dissecting pin and they'll be done by me. But again, they're used for anatomy when you want to hold open different tissue or even identification purposes when you're learning about arteries and veins and organs. There are often little tags with numbers on those. And you have to often, if you go into biology in college, identify those different organs, veins, and arteries. But again, we do most of those pieces virtually. These are little, these look just like little scissors that you'd use maybe like on your hair or eyebrows or something along those lines. But these scissors are a little bit, you can see right here, they have a little opening. And again, that's for dissections, but we don't use those. They're used to open up tissue. And again, if you were dealing with like a surgery, those also are the type of scissors that you would use to open up skin again, or cutting open tissue. A scalpel, we often substitute a razor blade instead of a scalpel. The scalpel itself has like a little attachment right here where you go ahead and you take the scalpel head out because you always want your scalpel to be really sharp. So you remove that head in order to exchange it for a new one when you're doing dissections. Usually use a new scalpel for almost every dissection or every other dissection because they get rusty, and you always want them to be extremely sharp. And they're usually surgical grade, so they are expensive. That's one reason why we don't use them too often. And of course, they're extremely sharp and accidents can happen. But again, that's used for dissecting and opening up tissue. These are probes. Again, a little hard to see, but when I show you in class, hopefully a little bit easier. There's a pointed probe where you can go ahead and move away tissue. Like let's say you're looking at one artery and it's laying on top of a vein. It allows you to go ahead and push that away. And this is to hold. So again, they're almost like extensions of your fingers. So when you're dissecting, you're not sticking your fingers in there. You might want to, that's fine, but sometimes it's even hard when you're looking at really, really fine tissue. You want to go ahead and have more of a fine exposure. Steps allow you to do that, and probes also allow you to hold it open. So microscope slides, often, so this part is where you usually write your specimen name. Often it'll be done for you, like a prepared slide will be labeled, and then you go ahead and then you put your specimen on the clear part. This is the part that'll be underneath the microscope. We're gonna have a whole unit just on microscopes. Should be the following week when we do cells and life processes, but it might be the week after. But you will all handle microscopes. So we use a little bit of the ruler when we were dealing with not micromeasurement, the observation lab. So when you're holding the specimen, remember you always hold it at the origin or the 0, and that will allow you to accurately measure your specimen. And we always use the metric side of the ruler. So if you're not sure, and that's quite common, even when I look at a ruler real quick, it's not common sense always. So just make sure the side that you're looking at actually has metric, right? So it'll either say cm or mm, or inches. So just make sure you're always looking at the metric side. measurements. We will be using goggles a couple times during lab, especially when we're heating liquids. You always have to wear goggles because the vapors, even though they may not feel like they go in your eyes, do, and they can irritate your eyes. So we always wear goggles, and sometimes even the chemicals that you don't see or smell can actually irritate. So we'll always be wearing goggles when we heat up liquids. So we will be measuring, I'm not sure if we're going to be using this particular balance. These are always used in high school science, but in real science lab, we never use their triple beam balance because it's too labor intensive. Even when you learn how to use them, it takes some time. We always use an electronic balance. So we'll most likely use those because in real lab, you never use, maybe I could be wrong, but the labs that I've worked at, you've never used triple beam balance. You always use electronic balance. And balances in biology tend to be pretty accurate. In chemistry, they're even more so, and they'll measure more effectively than a triple beam balance. Off the top of my head, maybe to the thousandth or tens of thousands of place for an electronic balance. And they actually will have like a glass cover so that even if the wind or dirt is going in from the air, it would actually measure that. So it could be completely enclosed, the electronic balance. And we'll be dealing with electronic balances. You'll see those in class. All right, measuring with graduated cylinders. So this is really important. This might be review for you. We'll probably have a worksheet or virtually you might have a worksheet for this. Depends on how class time runs. When you're looking at liquids, now a lot of people think it's just, this is always the case. But it happens to deal with a liquid that when you're measuring graduated cylinders, it depends on the liquid that you're using. So in biology, we usually use water. So that's represented as H2O. So this one obviously is water. We have something called a meniscus. Meniscus is, whoops. All right, now that all erased. The meniscus is, let's see if I can do that again. The meniscus is the curve piece right here, okay? And what you do when you're measuring is you actually avoid measuring this extra right here, right? So the little tip, because what happens is water is actually sticky or cohesive. So it sticks to the side of the glass. So in order to get an accurate measurement, what we use is the bottom of the curve here. All right, my drawing's not the perfect, but you get the idea. So you kind of ignore this piece and you ignore this piece, and you wind up with a measurement of, try to guess. You have notice you're going by twos here, right? So you got 6. So this would be 7, and that would be 8. So this would be 6, and then these are going to be 0.2, 0.4, 0.6, 0.8, right? And that would be 7. So it would be 6.2, 6.4, 6.6. So this one would measure 6.6, right? And most likely it's milliliters, right? But you wouldn't know that because it's not labeled. It is labeled on the graduated cylinder. So the meniscus itself is usually for water, which again is 99.9% of the biology. But when you're dealing with other liquids like oils, there will not be a meniscus. And sometimes you'll actually have a, what color? Well, we'll just use the same color. You actually have an opposite meniscus. I can't remember the type of liquids. This would be more of like a chemistry lab. So everyone always thinks there's a meniscus whenever you measure liquids. No, that's not true. It's only when you're measuring water-based liquids or mixtures, which again tends to be most of biology. But in chemistry, you wind up dealing with opposite because the liquid itself actually wants to stick to itself more than the glass. So you actually get an upside down one. So again, that's just kind of a misconception that has been taught over and over again. But if you ever work in a real science lab or you're dealing with chemistry lab next year, it's always good to know the reality of it, which is only water-based solutions are going to have that meniscus. All right, so this is really, really a common mistake. You never, ever, ever want to measure using a beaker. So there's taboos in science. One is using inches, and then the other one is measuring. Well, there's a lot, but in lab dealings, the other taboo is measuring with beakers. So a lot of students, and I get it, I would probably do the same thing. Is when you look at it, you see the milliliters right here. It says 150 milliliters. So you would think you'd measure, but this is just to kind of show how approximately how much liquid that this beaker can hold so that you don't pick up the wrong beaker. So let's say you're dealing with a graduated cylinder and it's 200 milliliter graduated cylinder, you would not pick this beaker because it would just overflow. So the numbers here are just really representative of what you can use when you're just grabbing a beaker from Science Lab. The truth is we wind up giving you beakers. You usually don't have to select, so it's kind of done for you. You don't really think about it too much. But in Science Lab, you need to know how much liquid you're going to need to hold in your particular beaker. So that's what the numbers are there, not for measuring. And it also tells you if you did a good job of measuring. If you said you grabbed 100 milliliters and it's overflowing, you did the wrong thing. So it's just there for a check. But you never measure using a beaker. It's just those increments are not accurate at all. And when you're dealing with Science Lab, a lot of people want to reproduce what you do. So if you're measuring wrong, it's like baking. If you use 4 eggs instead of 2 eggs, and then you try the cake and it's not good, you use too many eggs. So as a baker, you would never make that mistake. So same thing with science. You never make that mistake with using measurements in beakers, always in graduated cylinders. And always mixing. So the reason why you're using a beaker is for mixing liquids and or heating. Remember I told you to put that test tube inside a beaker filled with water. So that's how we always heat our test tubes. So microscopes, I'm not going to spend too much time because we'll have a whole unit just on those. This is pretty much what our microscope will look like. It's a compound, meaning it has 2. Compound means 2. So you have an eyepiece right here, and then you have your ocular lenses. So this is the most common microscope that we use in biology class. It's a light microscope, meaning its source is usually at this point in time a light. It used to be sunlight. Back in the day when it was first used, but now we actually use like an LED light. Before that was like an incandescent light. There are other type of microscopes we'll talk about in science class: scanning electron microscope, transmission electron microscope, and then we might even go into some other types of microscopes. Those are the ones that view viruses or inside cells, where this one is just going to view cells on the outside and the nucleus. So these are good for looking at bigger type specimens. But again, they have to be translucent, like see-through. Always when we're dealing with this, when we're dealing with heating of test tubes, if you're walking around the lab class, you never just want to hold the beaker, or I'm sorry, not the beaker, the test tube. You always want to have it in a test tube rack. So maybe this is the third taboo. You just never hold the test tube in your hand. You always want a test tube rack. Multiple reasons. One, it could just bump out of your hand and break. 2, it could be hot, and then when you're pulling it out of the hot bath, you break it that way. So we always use this for transportation, and plus you're usually having chemicals inside of your test tube, so you don't really want to spill it on yourself. This is just another safety feature. So we always use test tube racks. You'll be dealing with small pipettes. They're going to be usually plastic micropipettes, and if you look closely, there actually will be, again, like a measurement on there. You can use that for a measurement. It's usually, most micropipettes are between 1 and 2 milliliters, so this is going to be a small amount of liquid that you can use. Again, we're not going to be using that too much for measurement. But in the science lab, it is definitely valuable or useful. You don't want to flood your organism with too much water. This one we'll be using when we're dealing with electrogel and sorry, I said that backwards, gel electrophoresis. This is when we're doing DNA labs, and we'll be dealing with micropipettes. We'll be doing a demo, and then depending on how things go, we'll have one person per group, or maybe even have each individual, if we have enough micropipettes, to go ahead and practice. These are more common now with DNA and DNA analysis. They're quite often used in science labs, and they're going to measure in the microliters. So before we were talking about milliliters, there's something called a micro. Micro is 10 to the negative 6, which is quite small. So that's going to be hard for you to measure. That's why I have a little machine that does it for us. So we'll be using some filter paper in order to use a funnel. So a funnel usually I mean, I haven't used the one without it. Again, in biology, it's a little bit different than chem lab, but funnels are usually used for separation purposes. So usually what you do is either filter paper, which almost looks like a coffee filter paper, where it lets liquid come through and the solid stuff stay in, or cheesecloth, or something along those lines, where it actually filters out large particulates. And that's really important when we're dealing with biology, because sometimes if you want like the small stuff like proteins or DNA to flow through, then we go ahead and we filter it out from, let's say, the cellular components, which might be, just a cell membrane and thicker pieces of the cell, and they'll remain on top if we want to look at the smaller pieces of cells. So we use funnels quite often. A thermometer we will use a couple of times, probably not too much. The thermometer in a science lab is usually electronic probe. We don't use these anymore. It's not to say you can't. It's just the probes are just more accurate. Literally, it's just a temperature gauge. I mean, I'm sure you've seen it when people are scanning your temperature. Things have come a long, long way. But again, in high school science labs, these are cheap and easy to use, and you're going to go ahead and usually measure using Celsius, not Fahrenheit. So Celsius is the metric measurement for temperature in biology class. And again, it might change. Chemistry, you might be using Kelvin, though you won't be using a thermometer for that. But depending on what science you are in, depends on the units that you are focusing on. So Petri dish, you might have seen this when we did the observation. These are great when you're dealing with live specimens like insects, small things. We also use them for bacteria, but we're going to stay away from that this year. I think we have enough going on with viruses and illnesses, so we're not going to grow any bacteria. But I might have a sample that I can show you, just from the class perspective of what it would look like, and that's really important, because when you're finding out if you have strep or bacterial infections, that's actually what they use. It's kind of behind the scenes, and they pour a liquid in here, and they go ahead and then they do swab, that horrible swab that goes in the back of your mouth, and then they go ahead and then they just swab it, on this broth. It almost looks like Jello, like yellow Jello. And they go ahead and they swab it on there. And then what they do is when it grows, they go ahead and they see what's growing inside your mouth or what an infection. And they can go ahead and then give you the right antibiotic to help cure you. Because if you give the wrong antibiotic, it's not going to do anything. So they want to know the species of bacteria that's growing on you. So they grow it in this type of plate. Again, it's got Jello on it. So otherwise it won't. Or blood, depending on the type of bacteria. All right, so most of these are going to be simple tools that we're going to be using, and this is just a quick review for you to be familiar with. So make sure you filled out your worksheet. You can go back and look at it. Maybe some of these you're more familiar with. Pretty much everything should be in order, so it's easy for you to just go back and click on it. And we'll be reviewing this in class, and I'll be bringing the actual tools in class, and I'll be asking you for class credit for this assignment to go ahead and identify, but you'll have the answers right in front of you, right? And also if you feel comfortable talking about what that particular object is used for and how it's used for a lab. Okay? Thank you.